Digestion is a process which converts macromolecules into micromolecules. Digestion is basically a mechanochemical process. Mechanical action involves the grinding of food particles in the mouth as well as muscular grinding in the stomach. Chemical process refers to enzymatic action. Various digestive glands secretes their digestive juice, which contains many enzymes to bring about the process of digestion. Digestion starts in the mouth, and in the mouth, digestion occurs with the help of saliva, and this phase of digestion is called buccal digestion. Saliva contains one functional enzyme, that is salivary amylase, which is relatively weak enzyme. It can hydrolyze up to 30% of the starch. Starch is a glucan. It's a polymer of glucose, and it contains alpha-1,4 and alpha-1,6 glycosidic linkage. But salivary amylase breaks only alpha-1,4 glycosidic linkage. It cannot break 1,6 linkage. Therefore, a compound isomaltose is formed, which contains alpha-1,6 glycosidic linkage. In mouth, starch is converted into maltose, isomaltose, and limit dextrin. This is only 30% of the starch. After completion of buccal phase of digestion, food is called bolus. Now, bolus enters into stomach through esophagus. The only function of esophagus is to conduct the food particles from mouth to esophagus. Digestion, which occurs inside stomach with the help of gastric juice, which is a secretion of gastric gland, is called gastric phase of digestion. In the gastric phase of digestion, it mainly contains enzyme for digestion of proteins. One of the protein called casein, which is a milk protein, is acted upon by renin. Renin is not secreted in adults; it is secreted in children only. So renin is a milk coagulating enzyme rather than a milk digesting enzyme. It coagulates milk protein. How? First of all, renin acts on casein to convert them into paracasein, which precipitates with calcium ions to form calcium paracaseinate. This is in fact called, which is finally acted upon by pepsin, which converts them into peptones. Peptones are short peptide. Pepsin is the strongest proteolytic enzyme, and it is an example of endopeptidase enzyme. Endopeptidase enzyme are those which breaks a peptide bond from inside. Suppose this is a chain of polypeptide. If pepsin act on peptide bond from inside, so a complex is formed. This complex is called peptones. Other proteins present in the diet, proteins other than milk protein, are directly acted upon by pepsin, which converts them into peptones. After completion of gastric digestion, food is called chyme. Now, chyme enters into intestine. In intestine, it gets mixed with the secretion of various digestive juice like pancreatic juice, bile, as well as intestinal juice itself. First, we consider the action of pancreatic juice. Pancreatic juice is considered as complete digestive juice because it contains enzyme for the digestion of all the 4 biomolecules like proteins, carbohydrates, as well as nucleic acid and lipids. First, starch. 30% of starch digested in buccal cavity. Remaining 70% of the starch is digested by pancreatic amylase. It is like salivary amylase also breaks alpha-1,4 glycosidic linkage, but in alkaline medium. So, in starch is converted into maltose, isomaltose, and limit dextrin. Proteins which are not digested in stomach are now digested by trypsin and chymotrypsin of pancreatic juice. It is also an example of endopeptidase. Therefore, peptones are formed because it breaks a peptide bond from inside. So, the short polypeptide formed are called peptones. Now, some of the peptones are acted upon by carboxypeptidase enzyme. This is an example of exopeptidase enzyme. Exopeptidase breaks peptide bond from C terminal or N terminal. so that a dipeptide and amino acids are formed. Emulsified fat. Emulsification is the function of bile salts. Emulsified fats are acted upon by pancreatic lipase, which is also called as trypsin, which breaks them into their constituent, 3 fatty acid and glycerol. Almost all fats are digested by pancreatic lipase. It is the strongest lipolytic enzyme. Pancreatic juice also contain nucleases, which includes DNase and RNase, which acts on nucleic acid to convert them into nucleotides. Now see the action of intestinal juice. Intestinal juice, first, how does digestion of carbohydrate occurs by intestinal juice? Enzymes present in intestinal juice are mainly for oligosaccharides. It is maltase acts on maltose to convert them into glucose and glucose. Isomaltase on isomaltose to convert again into 2 molecules of glucose, because isomaltose and maltose are isomers, so constituent units are same. Lactase acts on lactose to convert them into glucose and galactose. Sucrase converts sucrose into glucose and fructose. Limit dextrinase converts limit dextrin into 2 to 6 molecules of glucose. For proteins, they have aminopeptidase, which is again an example of exopeptidase enzyme. It breaks peptide bond from N-terminal of a polypeptide and convert them into dipeptide and amino acids. Finally, dipeptide is acted upon by dipeptidase enzyme, which converts them into amino acids. Nucleic acid, which have already changed into nucleotides, are acted upon by nucleotidase to convert them into nucleoside and phosphate. Nucleosidase converts nucleoside into base and sugar. Now, after completion of the process of digestion, which is completed in a small intestine, food particles are absorbed. Absorption, the chief site of absorption is small intestine. Theoretically, absorption does not occur in mouth. Little bit absorption of some small amount of water and alcohol, certain drugs like aspirin may occur in stomach. However, most of the part is absorbed in small intestine. The wall of small intestine is provided with villi, which contains 2 types of vessel. One is called blood vessel, other is called lymph vessel. Fats and fat soluble substance are absorbed in lymph vessel. Any other substance is absorbed in blood vessel. Now, how does absorption of fats occurs in small intestine? The end product of fats is fatty acid and glycerol. The fatty acids are water insoluble substance, therefore it cannot be absorbed as such. Therefore, they form an aggregate with bile salt called micelle, which is a water soluble compound. It is absorbed by the intestinal villi. By simple diffusion. Fatty acid, glycerol again unites to form fats. In the villi, the shell of villi, fats are coated by a molecule of protein. Now the complex becomes lipoprotein. This lipoprotein complex is called chylomicron. Now chylomicron is thrown into lacteal vessel, which is a lymph vessel, by a process exocytosis. Chylomicron is also water soluble substance. Now lacteal vessel, all the lymph vessel finally opens into blood vessel. So from chylomicron from lymph vessel enters into blood vessel. In blood vessel, certain enzyme present in blood plasma breaks the protein part of chylomicron, so the compound again becomes fats. This fats gets deposited in adipose tissue.